Hi everyone, welcome back. So all living things and some non-living things like viruses have some kind of genetic material, DNA or RNA. And biologists across a variety of fields, including medicine, ecology, evolution, and more, have to study that genetic material in order to better understand that organism. Now, one of the first steps, oftentimes, to studying the genetics of any living thing is to extract its DNA, isolating that DNA from the organism that it came from and separating it from any other biological material, these other molecules like lipids and proteins and carbohydrates, things that might interfere with our ability to analyze that DNA if they're all clumped together. Now, once we have isolated that DNA, we can do a bunch of different things. We can learn a lot about the organism, and in particular, the proteins that it makes. And by learning about the proteins that it has the ability to produce, we can also learn about its metabolic ability. Those proteins, a lot of them are going to be enzymes and have enzymatic activity. And so we're going to learn a lot about what kinds of materials it can produce, what kinds of things it can do with those enzymes. We can also learn a lot about the ancestry of that organism, who it descends from, and who it is more or less related to. And today, with modern technology, we can do even more exciting things, like potentially isolate specific genes and alter the genetics, the DNA, of that organism or another. We can take a gene from one organism and insert it into another so that that organism produces proteins it doesn't usually make. When we go to do an extraction, there are a variety of methods that we can use, and they range a lot in their complexity. Now, which method we use really depends on the organism and the sample. There are a variety of conditions where you'd want to use a more complicated or more specific extraction protocol, but a lot of these methods are pretty simple as well. Importantly, extraction isn't always successful when we go to do it. Like anything, it's a lab method that has the propensity for error. Human error is a common thing, whether we like it or not, and there can be a variety of unexpected issues that pop up during the process that make it so that we don't end up actually isolating DNA. An important additional step to this research is to have a method that helps us make sure that we have successfully extracted DNA. How can we tell after we have done a DNA extraction that we actually have enough material to do the rest of our research with? This is a really common problem. Now, researchers use DNA in a variety of ways, including taking pieces of the DNA and amplifying it, making many copies of that DNA. In some cases, researchers will be cutting up that DNA at specific locations. They might be inserting new pieces of DNA, doing gene editing, or deleting regions of DNA. We also need some kind of method to test and make sure that when we have done some of these applications, we've made copies or inserted things or deleted things, that the method actually worked. How can we tell that the manipulations that we did to DNA actually happened? After all, we can't literally see the molecule. It's far too small to see even with a microscope. This is where gel electrophoresis comes in. This is a near universal and essential tool in genetics research, and a lot of labs use this in one form or another. It's a method that, first, observes the presence of DNA after an extraction has been performed. It helps us actually visualize DNA in large quantities, even if the molecules themselves individually are too small to be seen with the naked eye. It will do a really important job of sorting DNA by the size of the molecules. This is a really useful method for making sure that if we, say for example, had to cut up a bunch of pieces of DNA into specific sizes, or add a piece of DNA of a specific size to a plasmid or something like that, we could look to see if the DNA is the specific size that we intended it to be. That will be an indicator as to whether or not this method that we used actually worked. And so it can be a great diagnostic tool to assess if our methods in other research was successful. So we find gel electrophoresis often paired up with other study methods. So now we're going to try to understand how gel electrophoresis works, but to do that, we have to re-examine the structure of DNA. So remember that DNA is a very long chain of linked up molecules. It's a polymer of nucleotides. And there are two of these strands that are connected at the middle and twist into that double helix shape that we're familiar with. Now, the backbone of DNA on either side of this molecule is the sugar phosphate backbone. It is made up of sugars and phosphates. And an important thing to remember about phosphates is that they have a negative charge to them. That will be relevant in a minute. So to study DNA through gel electrophoresis, we're going to load it into a porous gel medium. This is something that's pretty familiar to us. We're going to use agarose, the same stuff that we use to give solid medium that we find in petri dishes, the same kind of gel-like consistency. Now this gel has a lot of microscopic pores. Now DNA is going to be able to move through the spaces within this medium. However, the larger the DNA molecule is, the slower it will move. Now let's take another look at that DNA molecule one more time, right? So we can see those specific phosphate groups. Now as a reminder, those phosphate groups have that negative charge. These are the secret to gel electrophoresis. Now, other parts of the DNA molecule normally don't have that significant of a charge. However, all of these phosphate groups have that slight negative charge to them. This comes from a single oxygen that we find on every single one of these phosphate groups that keeps an extra electron. For living things, this charge that phosphate groups have is very helpful because water loves to interact with partially charged or polar molecules, things that have some kind of charge to them. And as we know, our cells are mostly made of water. The vast majority of the volume of our cells is liquid. So the fact that DNA has a negative charge actually allows DNA to mingle with water. It allows it to become dissolved in water. This is really valuable because it allows DNA to be manipulatable. It allows us to actually utilize DNA as a storage of information, effectively a massive library of protein instructions that we can go back to at any time to look at how to make this or that specific protein. Now this quality, the negative charge to DNA, is also essential for gel electrophoresis. This is because if we send an electric current through that gel when DNA is in it, the negatively charged DNA is going to be attracted to and move towards the positively charged side of that electrical current. DNA is always going to move in a uniform direction from one side to the other side. And because molecule size determines the travel speed, we're going to find that smaller molecules are going to travel farther through this gel than larger ones. So the larger the molecule, the more close it is to the starting point. The smaller the molecule is, the further it will have traveled. It's also extremely predictable and measurable. We can actually measure how large a molecule is by how far it's traveled through the gel relative to other molecules. This is what this process looks like. So in a gel, as we run the gel, you're going to see that different size molecules travel at different speeds. And so we're going to have bands that form of specifically sized molecules that if you have a lot of one specific size, they're going to group out together like this. This is basically what is happening inside that gel. So larger molecules are tumbling through these tiny pores in the gel, but because they're so large, they can get tangled up a lot in different places, and so they are slower to move along that path. However, smaller molecules, because they are simply smaller, they get tangled up less, they fit more easily through these different pores, and so they travel faster. When we run a gel, we usually stain the DNA with some kind of chemical beforehand so that we can visualize how far it travels. We use usually a fluorescent tag that we can stick onto the DNA molecules. And then we can put the whole gel under a UV light box, and that will make the fluorescent tags on the DNA glow. This helps us actually visualize where this stuff fell out on the gel. Now, when we do this method, control samples are really important. We use something called a DNA ladder. This is a sample of DNA where all of the fragments are of a known size. So this is a really important thing that we usually see on the left-hand side of every gel. And it is essentially a benchmark so that when you look at your samples, you can look back at this benchmark and say, well, it falls between this molecule and that molecule. And I know that this molecule has a size of 1500 base pair and the one below it has a size of 1000 base pair. So my molecule must be between those two measurements. This is the value of a DNA ladder.